Hello everyone. Good evening. Just wait for 5 to 10 minutes, then we'll start our session. Hello everyone, good evening. Am I audible to you and my screen is visible? Please anyone
Yes,
comment.
ma'am.
Okay, thank you. So this is Subhasmita from IIT Indore. I am a PhD scholar here in second year and part of PMRF TA work I am doing this NPTEL TA session and today we'll be talking about some questions or assignments regarding the week 2 lecture series, that is breakage and repair of genomic DNA. Okay, let's start then. Transposition is any mutation where a purine base is replaced with a pyrimidine or a pyrimidine is replaced with a purine. This is option A. Second option is a purine base is replaced with another purine. Third option, a pyrimidine is replaced with another pyrimidine. And fourth option is a purine or pyrimidine base is deleted. So please try to answer in 2 minutes. Then we will discuss the answer and all the theory behind it. So many of you have answered A. So let's talk about the theory and what will be the answer. Let's see. So first thing is mutation. What is mutation? Can anyone reply please in the chat box? What is mutation? Yeah, anyone? Okay, change of a base. Yes, it can be a change of a base. If we talk about a single base changing, then it will be called as a point mutation. Because the changing of a single base or single nucleotide would be called as point mutation. Just we can say what is mutation? The change in the sequence of DNA. Okay, change in sequence of DNA. In case of eukaryotic and other higher organisms, DNA is the genetic material and change is the sequence is called mutation. Just a second. And so there are types of mutations like chromosomal alterations can be happen or point mutation can happen. Point mutation, there will be change of a single base, or if there is some chromosomal alteration, then it might be deletion, insertion, and duplication also. These all are called as chromosomal alteration that leads to a different protein product, so that a different function will be there. Okay, so mutation can be harmful also and it is blessed also. Why it is helping us? Because of this evolution. Okay, so because of mutation we get new allele, new trait, and evolution can happen throughout this long decades we have seen how humans have been evolved and how different prokaryotes to eukaryotic organisms have been evolved. This has been happened because of this mutation only. Sometimes this mutation leads to harmful causes because of any protein changes, and that might lead to any disease or any kind of abnormalities. So move to our question, that is what is transition and transversion. So this transition and transversion, basically it is a change of a single base or nucleotide. That's why it's called a point mutation. Okay, so transition is when a purine nucleotide is changed to another purine, or a pyrimidine nucleotide is changed into another pyrimidine. What are purines? Adenine, guanine, they all are purines, while cytosine and thymine are pyrimidines. Right? Just a second. Okay. So if you have any doubt regarding this, then you can question me in the chat box. Okay, you can ask me anytime. So here it is a adenine, guanine. They are purines. This is a simple representation of transition and transversion. They are cytosine and thymine. So if adenine is changed to guanine, if or guanine changed to adenine, that means if the nucleotide base mutation happens in the purines only, in between the purines only, that will be called as transitions. If adenine may be converted to thymine or cytosine because it has been changed to a pyrimidine, it will be called as transversion. Or same as you can see in this simple schematic representation. Transversions are any mutations in which a purine is replaced with a pyrimidine or vice versa. And these transversions are more likely to change the amino acid sequence of proteins than transitions. Why? Why it is so? Anyone? Here it is written that transversions are more likely to change the amino acid sequence of proteins than transitions. Okay, just let me answer then. So transversions, what happens? This adenine changes to, might be changes to thymine or cytosine. Then it will be binding with G or C, like this. So that may be changes our the whole frame, the reading frame, and the sequence will be changed, and that's why it leads to a different protein product for all the cases. And that's why transversions are more likely to change in the amino acid sequence, because it's just like acts like a frame shift mutation and changes the whole reading frame. Okay, understood? Fine, I guess everyone is getting my point, what I'm trying to say. Okay, then I think transition and transversions are clear for everyone. Let's move to our next question. That is, the key function of DNA topoisomerases is to replicate DNA, to synthesize mRNA, to synthesize protein, to relax superhelical DNA. Please answer in 2 minutes, then we will discuss. Okay? As many of you have answered, let's discuss. Okay. So the key function of DNA topoisomerase is to relax superhelical DNA. As many of you answered D, very good. You are quite brilliant students, I guess. You are giving correct answers. So let's discuss a little bit about DNA topoisomerases, how it functions and how it's doing its work. Just a second. So I guess you are able to watch my screen. So just keep attention. This DNA replication process, it's a, I think you might have read about this DNA replication process, and there is one lagging strand and there is one leading strand. And what this topoisomerase does, it just opens up the DNA. As you know that DNA, this parent DNA, okay, this is the parent DNA, it is a double helical structures DNA. So topoisomerase just opens up the double helical structures and create each and every strand as a template for DNA replication process. You must have heard about the word semi-conservative DNA replication process. That means this one strand of each DNA double helix might act as a template for producing the new strand of DNA. So here the enzyme DNA polymerase, and it is the Okazaki fragments. So where the Okazaki fragments generally found, it is in the lagging strand. We can see the Okazaki fragments, and it's the RNA primer required, and primase, helicase. These are the enzymes required for the DNA replication process, and this is the leading strand. So there will be no Okazaki fragments, and topoisomerases generally helps in opening up the DNA. So DNA topoisomerases are family of essential enzymes abundantly present in both prokaryotes and eukaryotes. For example, in human, seven top genes are found, including top 1, 2, 3, and etc. They can act as an endogenous source of DNA damage. So what is an endogenous source? Endogenous means what? Inside. Okay. So when a cause is there and it is damaging the DNA from inside the cell only, that's called an endogenous DNA damaging factor, and topoisomerase act as another endogenous source of DNA damage, and its principal function is to relax superhelical DNA during DNA replication and transcription. Okay. So in last class we discussed about this prokaryote and eukaryotic chromosomes. So can you please tell me where this transcription and replication happen in the prokaryotes? In prokaryotes, there must be also DNA replication, transcription, translation also happening, right? So uh, as I know and you also know that there is no nucleus in the prokaryote. So what is the place or where this DNA replication, transcription, translation is happening inside the prokaryotic organism? Chromosome is not a place to uh, just happening these processes. Okay, chromosome is what? Chromosome is the compact structure of DNA. It only contains the DNA along with histone proteins in case of eukaryotes and other proteins in case of prokaryotes, right? So chromosome is just the compact structure of DNA. As you know that DNA is a long chain of this nucleotide bases, right? Sweta and Sivangi, cytoplasm, yes, it's true. It's cytoplasm. And I'm just telling you the chromosome thing, right? Chromosome is nothing but the compact structure of DNA where DNA is present along with different proteins. Understood? And it is not a organelle or any environment for this replication and transcription process. Okay, as you have already answered cytoplasm, it's great because in cytoplasm only there is one place, I can say, at certain part this DNA compacts up, or the genetic material of the whole organism compacts up at one place, and it is called nucleoid, right? We just discussed this thing in our last class. This is nucleoid. Nucleoid, at that place this DNA is compact in shape, and at that place is the responsible for DNA replication and transcription process. As we know that for translation ribosomes are required, so translation happens in the cytoplasm only, and we can say that transcription and translation happens simultaneously, not like in eukaryotic organisms. Eukaryotic organisms, first there will be replication, then there will be transcription, and then there will be translation. Okay, but in case of prokaryotic organisms, all the things happen simultaneously in the cytoplasm only. I'm talking about these things because sometimes basic need to be clear, right? We need to clear our concepts regarding these things, then only we can solve tough questions. Okay, then uh, let's move to our next question, that is, deamination of adenine produces hypoxanthine, xanthine, uracil, or thymine. Please answer in 2 minutes, then we will discuss. Okay. Okay, so many of you answered A, and it is the correct answer. Great. You are answering correct ones only. That's great. And other people, other students, I will highly suggest you to please answer the questions. Just try, okay? If it is wrong, then it's wrong. No need to worry that if I get wrong, I will be scolded like this. Okay, just answer. So deamination of adenine produces hypoxanthine. How it produces hypoxanthine? Let's look. So this is the structure of adenine, right? And deamination. This word suggests that one ammonia group is produced from this, and deamination means it is removed, right? So one ammonia group, that is NH3 plus, has been removed from this adenine, and this is called deamination process. So how this deamination happens? It is generally happens spontaneously in the presence of water. Okay, in the presence of water molecule. Anyone wants to say anything? Okay. So in the presence of water molecule, here, this, it is NH2. If we add one water molecule here, then the H plus and OH minus will be produced, and this H plus along with this NH2, it will be removed as NH3 plus, and OH will be bound to that part. Okay, OH, here it will be OH, and OH, it this pi bond will move to at this part. Pi bond will move to at this part, and this OH will produce one H plus ion and becomes a double bond structure. Here it will be an acceptor. The whole process, it will be 3 or 4 step process, but I have not written long processes because no, biology doesn't need that much of chemistry, but it will be great if you know these things. Okay. Again, I am saying, just repeating what I said. This is the structure of adenine. Just let me write if I can. Uh, okay. So here one NH3 plus will be removed. Okay. One NH3 plus will be removed in presence of water. In presence of water, one NH3 plus will be removed, because water, if we add water, it will what will the ionization form H plus and OH minus. Then it will the H plus will bind to this and NH2, and it will be removed as NH3 plus, and then OH will be bound this at this point, and after this pi bond will move to this region, and the H will be removed as H plus ion, and a double bond will be formed, and this structure will be known as hypoxanthine. In some cases, hypoxanthine structure is shown as single bond OH. Okay. In some cases, it will be single bond OH, but in case of uh, uh, this figure, it is written as double bond O. In case of guanine, there will be no deamination process. If we don't do that, then it will be xanthine. So A stands for the hydrogen bond acceptor, and this stands for the hydrogen bond donor. Okay, I guess it's clear. Now adenine to hypoxanthine and guanine to xanthine. Okay, let's move to our next question. That is, which of the below is an endogenous DNA damaging factor? Options are replication errors, ionizing radiation, ultraviolet radiation, environmental stress. Please answer in 2 minutes. Then we'll be discussing this question. Okay. Hello, am I audible? Hello, am I audible? Hello. Sorry, guys, there was some network interruption actually. Uh, the Wi-Fi is so unstable. That's why I just got disconnected. Thank you for waiting. Are you able to listen to me? Okay, thank you. Thank you so much. Thank you so much for waiting. Actually, the Wi-Fi just so down and it just disconnected. Okay, let's start. Okay. So is my screen visible to all of you, and my am I audible? Please let me know when you can't hear me or you can see the screen. Okay. So the question was, which of the below is an endogenous DNA damaging factor? And the answer is A, that is replication errors. Okay. Some of you have answered C or D, so I'll talk about it. What is the answer, like what is endogenous DNA damaging factor and what is exogenous DNA damaging factor? Let's talk about it. So there are certain factors that damages the DNA. So what is the meaning of DNA damage? DNA damage means certain part of DNA or certain base, some part of the DNA is broken. As you can see here in the figure, see here in the figure that certain part of this DNA is broken. It's called DNA damage. So how this happens? This might be due to some endogenous factors or might be due to some exogenous factors. Endogenous means the factors are from inside the cell, and exogenous means the outside factors, outside the living body or outside the cell. Okay, so endogenous factor includes replication errors. Replication error means when the DNA replication happens in the S phase of cell division. At that point, if some error happens during a replication, that's known as endogenous factors, that because that's been happening due to some error mechanism present inside the cell. Okay. And DNA base mismatches. There might be some DNA base mismatches, like if G is pairing with guanine is pairing with cytosine, so it in place of cytosine, if there is a base like any type of A or T, so that will be called DNA base mismatches, and that occurs due to some error mechanism inside the cell only. That's why it's called endogenous factor. It might be due to topoisomerase DNA complexes. If topoisomerase DNA complex is strong and topoisomerase can't open the DNA double helical structure properly, or it might some there might be some problem during the replication, or spontaneous base deamination, that just we discussed about adenine converting into hypoxanthine, like that. A basic site means a purinic site or a pyrimidine site. That means there will not be any base. Okay, so at that point the structure will look like this, that no base will be there. That's called a basic site. Okay. Oxidative DNA damage, because of oxidation, if DNA is broken or some part of this phosphodiester bond is broken, or hydrogen bond is broken, so that's called oxidative DNA damage. DNA methylation, as you all aware of DNA methylation, it is known as one epigenetic modification. Methylation means adding of methyl group. Okay, so adding a methyl group leads to certain changes in the DNA, and that because of that, if a mutation happens or DNA damage happens, that's is called endogenous factors. Okay, next come to exogenous factors. Exogenous factors might be ionizing radiation, like different radiation which harms the DNA, or ultraviolet radiation, alkylating agents, aromatic amines, polycyclic aromatic hydrocarbon, reactive electrophiles, toxins, environmental stress, or like this. Okay, so exogenous factors might be due to some ultraviolet radiation damage. Alkylating agents also can damage the DNA, and reactive electrophile toxin, like this. Okay, I guess it is clear to everyone. Please let me know if you have any question. Let's move to our next question, that is photoreactivation repairs UV induced DNA damage, replication errors, interstrand crosslinks, or abasic sites. Please answer within 2 minutes, then we will discuss. Okay, please answer. Try to answer. 42 will be. Oh, at this time I was not audible. I talked a lot. Sorry. Okay, 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 fine, fine. Am my screen visible? Okay, okay, okay. Okay. Is it visible now? Okay, thank you, thank you. Thank you, Virendra. Okay. So I was talking about this endogenous and exogenous DNA damaging factors. Okay. So endogenous DNA damaging factors might be replication errors, DNA base mismatches. Replication errors generally happen during the replication of DNA in the S phase of cell division, right? S phase of cell division is responsible for DNA replication. At that point, if certain uh, error mechanism is there which leads to this DNA damage, that will called as replication errors, and it may be DNA base mismatches, like G, if G pairs up with T or A, like this, or topoisomerase DNA complexes. If the topoisomerase is not able to open up the double helical structure, and sometimes it is a strong complex, and that might be leads to some DNA damage. Spontaneous base deamination, like we discussed just before, like adenine is been deaminated to produce hypoxanthine, or production of A basic sites, like your A pyrimidine site or A purinic sites called A basic sites. This A basic sites will look like this, like there will not be any base, any nucleotide base, that will be called as A basic site. Oxidative DNA damage, that might happen due to oxidation of DNA or single base pair. DNA methylation, as you know, it is one of the epigenetic modification. If DNA methylation leads to any DNA damage, then these all factors are happening because of the cell error mechanism only, inside the cell only. That's why it's called endogenous factors. If we move to exogenous factors, it might be ionizing radiation, ultraviolet radiation. How ultraviolet damages the DNA and how it will be repaired, we'll be discussing these things in our next slide. And alkylic agents, aromatic amines, or polycyclic aromatic hydrocarbon, reactive electrophiles, toxins, environmental stress, they are coming out from outside, not inside the cell. That's why it is called exogenous factor. Okay, understood? Are you getting my point? What are the difference between endogenous factor and exogenous factor? Okay. I think you are trying to understand. Let's move to our next question, that is photoreactivation repairs UV induced DNA damage, replication errors, interstrand crosslinks, or abasic sites. Please try to answer, then we will discuss. Here it is. Question. Is it visible now? Okay, thank you. Thank you for confirming. Yes, great. You are answering correct. So the answer is UV induced DNA damage. Okay. So photoreactivation repairs the UV induced DNA damage. Or so we'll talk about it. Okay. So this photoreactivation is the process by which UV inactivated organisms restore their function by repairing UV-induced DNA damages with the energy of near UV light region, that is 310 to 480 nanometer, and an enzyme called photolyase. Okay, so remember this enzyme, it is generally asked in the simple questions in NET B section or GATE life science or biotechnology questions. Okay, so this DNA photolyase is responsible for the repair mechanism of photoreactivation damage or UV induced damage, right? So UV photolesions and alkylated bases are two types of DNA lesions that can be easily reversed in an error-free way in lower organisms and mammals through photolyase mediated photoreactivation of UV lesions. How it happens? Just look at this figure. Here, one lesion has been formed because of this UV induced damage. So generally, because of UV, ultraviolet rays, DNA damage happens. And how it happens? Can you please tell me what is the term called if UV or ultraviolet is damaging the DNA, then what might be happening at here? There might be one thymine dimer formation. Okay. Please let me know if it breaks again. So this thymine dimer, what it does, because of ultraviolet this DNA damage happens and thymine dimer forms. It is called a DNA lesion. And someone is trying to join. Okay. And this photolysis binds this lesion, as you can see, and this happens in the visible light range near UV light region, that is 310 to 480 nanometer range. Try to remember this range, that is 310 to 480 nanometer, and this photolysis binds the CPD lesion and removes it, and photoreactivated photolysis cleaves the CPD, reverting to monomeric DNA. What is CPD? What is, how it is happening? Another question will be there, and we'll discuss it later. Just look at the schematic representation and try to understand how this photolysis is helping in photo reactivation. Next, let's move to our next question, that is bulky DNA lesions that we were talking about in the previous slide. Bulky DNA lesions such as cyclobutane pyrimidine dimers. Okay, it is called CPD. It might be thymine dimers generally forms because of ultraviolet light. And 6,4 photoproducts are repaired by base excision repair, mismatch repair, nucleotide excision repair, or photoreactivation. Please try to answer this question, then we'll discuss more about this photo reactivation and how it just repairs the UV induced damage. Okay, let's discuss what will be the answer. Please let me know if I am not audible or if my screen is not visible to all of you in the chat box. Okay. Okay, so this is a flow diagram, you can say, that is how UV induced damage is happening and how UVA UVB is damaging the DNA and how it can be repaired. Okay, though, so UV induced damage happens generally because of this UVA or UVB. UVA by oxidative stress and immunosuppression, and this oxidative stress leads to production of DNA photoproducts. Okay, what it produces? 8 oxo decoalations. Is my screen is not visible to you? I have already shared my screen. Okay, thank you for confirming. So this UV induced damage, it is because of this UVA and UVB. UVA leads to production of DNA protoproducts, that is 8 oxo decoalations, and UVB generally leads to production of photoproducts. So as you can see here, this DNA photoproducts, that is CPD, 80% to 90%, and 6,4-PP, that is 10% to 20%. They leads to mutagenesis, and this failure of apoptosis leads happen, and that leads to death of the cell. So as we previously discussed in the previous slide, as you can see here, it is written here in CPD. CPD stands for cyclobutane pyrimidine dimer, and 6,4 photoproducts that are generally produced by this UVB mediated DNA damage. And here this photoreactivation process is helping in removing these lesions in the DNA. So in with the help of enzyme DNA photolase. Okay, this DNA photolase helps in removing these large lesions in the DNA, that is CPD cyclobutane pyrimidine dimers, and removes it just in the visible range, and DNA ligase then ligase this strands to produce a general normal DNA. Okay, so I think you got your answer. Okay, fine. Let's discuss more about this nucleotide excision repair, mismatch repair, Beck's excision repairs in our following slides. Okay. Uh, this is our next question, that is xeroderma pigmentosum is a rare autosomal recessive genodermatosis caused by mutations in DNA replication mechanism, nucleotide excision repair mechanism, transcription, base excision repair mechanism. How this disease, geroderma pigmentosum, happens? Rare autosomal recessive. Try to answer, please. I think my screen is visible to all of you. I have changed the slide. Here it is the question. I have changed the slide. Can anyone see the question? Okay, thank you. Others, please try to answer. I think some of you have answered B, some of you answered D. These are general simple questions, not complex ones. Okay, let's discuss then what will be the answer. So xeroderma pigmentism is a rare autosomal recessive xenodermatosis caused by mutation in the nucleotide excision repair mechanism. So the answer will be B. Some of you have answered A, some of you have answered B or D. So answer will be B, that is nucleotide excision repair mechanism. Okay. And severe photosensitivity resulting in sunburn, skin pigmentary changes, malignant tumor formation, and occasionally progressive neurologic deterioration occur in this syndrome. It's not a disease, it's a syndrome. Okay, so I guess everyone has got your answer. We will be discussing this nucleotide excision repair mechanism in our upcoming slides. So, okay, this is our next question. In DNA photolyase, reduced flavin adenine dinucleotide FADH minus works as catalytic cofactor, light harvesting cofactor, metal ion, or antenna molecule. As you have previously discussed in our questions like on photoreactivation, where we found that DNA photolyase in prokaryotic and eukaryotic organisms is responsible for repair mechanism of UV induced DNA damage. So here another question is there, like if in that case FADH, this energy molecule, has been used by the mechanism, so it is asked us if it acts as a catalytic factor or light harvesting cofactor, metal ion, or antenna molecule. Please try to answer. Okay, let's discuss. In DNA photolysis, reduced is a monomeric flavin dependent repair enzyme with 422 616 amino acid residues. It contains two known cofactors, that is a catalytic cofactor and a light harvesting cofactor. Okay. Reduced flavin adenine dinucleotide work as the catalytic cofactor in all the photolysates. So the answer will be A. Some of you have answered B. So the answer is A, that is catalytic cofactor. It transfers energy to the cyclobutane pyrimidine dimers in the form of an electron, breaking the cyclobutane ring and producing two monomeric bases. So generally what happens when UV light damages the DNA, there this dimer formation happens. So dimer means two bases are joined together. So this flavin adenine dinucleotide, it provides one electron and breaks the cyclobutane ring to produce single monomeric bases. Okay, and this light harvesting cofactor includes MTHF, 8-HDF, FMN. They absorb light energy and transfer them to FADH. Understood? I guess you can correlate with this photosynthesis, because there are also light harvesting factors in the chloroplast and how it is happening. So here this light harvesting cofactors, the absorb light and transfer to FADH molecule. So it becomes reduced to produce FADH minus, and this FADH minus transfers one electron to cyclobutane pyrimidine dimer. Cyclobutane pyrimidine dimer means two bases are joined together. Okay, and it breaks the phosphodiester bond between them and produces single monomeric bases. Understood? How this DNA photolysase works, it's written here. I guess everyone is clear about this DNA photolysis, how it works, because we have discussed about 2 to 3 questions on photoreactivation and DNA photolysis. Okay. Okay, fine. I think everyone is clear about it. If you have any doubt, please ask me. We'll talk about it. Okay, let's move to our next question. That if the mutation has a negligible effect on the function of a gene, it is known as silent mutation, prem-shift mutation, substitution mutation, or insertion mutation. I think my slide is visible. Okay, thank you. Thank you for confirming. Okay, others please try to answer. This is an, I think it's a simple question as comparison to other MCQs we have attended. Okay, okay, let's discuss. All of you have answered A and very good. It's the correct answer. If the mutation has negligible effect on the function of a gene, it is known as silent mutation. So how it works? This mutation, I already discussed about what is mutation in the starting of this class. So let me repeat again. Mutation is nothing but change of DNA sequence. In the if the DNA sequence is changed because of any single point mutation in the base, it might be a point mutation or a chromosomal shift, chromosomal alteration. This chromosomal alterations can be like deletion, insertion, inversion, substitution. Okay, and point mutations, there are maybe transitions or transversions. Okay, just have a look on this schematic presentation, and just let me tell you about more about this mutation. Mutation classes and types of mutation: gene or point mutation, chromosomal mutation, spontaneous mutation, induced mutation. Okay, sometimes we follow this induced mutation in our labs also for our research purpose. Gene or point mutation, it is a change in a single gene or a nucleotide pair. Okay, this may be due substitution, insertion, deletion, or inversion. And chromosomal mutation, a change in a gene due to alteration of chromosome structure or number. Chromosomal structure can be changes as deletion, means some part of the chromosome is deleted. Okay, if a chromosome is there, because all of you know that there is 2 copies of each gene present inside the human, and if one chromosome, uh, some part of the chromosome is deleted, then that particular protein is will not getting formed, so that function will not be happening, and that's why it leads to mutation or abnormal processes. Inversion or translocation means translocation of certain, during crossing over, if the certain parts of chromosome A is transferred to certain parts of chromosome B, it's called translocation. Or duplication means the same thing is duplicated inside the chromosome only. Okay, this leads to overproduction of the protein. Okay, and chromosomal number, aneuploidy, polyploidy, these are considered as chromosomal mutation. Okay, In case of spontaneous mutation, it occurs spontaneously, that non-disjunction during meiosis or DNA replication. Non-disjunction means what? Means during meiosis what happens in the cell division phase is there, a mitotic phase is there, and S phase is there. So if in the S phase DNA replication happens, and if the non-disjunction happens in the chromosome, that may lead to spontaneous mutation. Induced mutation can be done using any mutagen. Mutagen, for example, are exogenous factors like we discussed in the previous slide. This might maybe an ionizing radiation or UV light or any alkylating agents or anything which is a mutagen. We can use that thing to produce an induced mutation at a particular site. Okay, I think it's clear. In the previous thing, we talked about this silent mutation. If this mutation has negligible effect, how it becomes negligible effect? So as you all know that this there is one triplet genetic code is present, and this responsible for a single amino acid inside human or other eukaryotic organisms, and it is conjuged, right? So if I write AUG, that is for methionine. Okay, just if I write AUG, okay, I think everyone is know this triplet genetic code. AUG stands for methionine. If this uh, mutation happens at guanine, that this third base of this triplet genetic code, this might lead to a U T. Okay, so just think of it as it is like it is this G is this guanine is mutated to thymine. Okay, so then only also it will produce this amino acid methionine, because in the last base of the triplet genetic code is known as the oval base. WO, sorry, it's the writing is too bad. Oval base pair. Okay, that means this third nucleotide base of triplet genetic code doesn't matter if we change it into a another nucleotide. Then only it also produce the methionine amino acid. So it will not create any changes in the protein or any function of this protein. That's why it's called silent mutation. I think everyone understood this thing, why it is called silent mutation. If any mutation happens at this third genetic code, that will lead to production of silent mutation, because the amino acid will be the same, okay, as it is for before. Frame shift mutation, what is it? Let's for example A G T C A. Okay, for example, this is our 5 dash to 3 dash strand of genetic code. Let's start T. Okay, if I insert one nucleotide base here, just for example it is C. Okay, if I insert C here, then the genetic code will be, the triplet genetic code will be A, it will be read as AGC, then TCA, and the T will remain like this. Are you getting my point? It is 5 dash strand to 3 dash strand. Okay, the triplet genetic this sequence is AZTCAT. This is the 5 dash to 3 dash DNA sequence. If I insert another nucleotide base C here at this point, so it will, the reading frame will change. Okay, before it will be read as AZT or CAT. It will be read as AZT and CAT. Now it will be read as AGC and TCA. So this TCA will produce another amino acid which leads to change of function in the protein. Protein function will change. It will lead to a different protein, or the function might be harmful to the organism. So this type of mutation is called frame shift mutation. Because of insertion or deletion of a single nucleotide base, this reading frame has been changed for the whole sequence, that is 5' to 3' DNA sequence. And different protein or amino acid has been formed by this mutation, and it's called frame shift mutation because the reading frame has been changed. I think everyone is clear. Substitution mutation means if a single nucleotide is been substituted with another nucleotide. It's called substitution mutation. Okay. Insertion, for example, you can take it as an example for insertion mutation because it is inserted. I think everyone is clear with silent mutation, frame shift mutation, substitution mutation, and insertion mutation. Okay. Thank you. Okay. Let's move to our next question. That is, which of the following mechanism will remove uracil and incorporate the correct base? We are talking about a single nucleotide, that is uracil, and incorporate the correct base. What will be the answer? Direct repair, base excision repair, mismatch repair, or nucleotide excision repair? What it will be? Please try to answer. I think everyone is getting the hang of what we are talking about, what is the DNA damage, how it happens, and how it is repair, the how repair mechanism works. So just try to answer. We'll discuss. Question not visible. Okay, slide not changed. I have changed the slide. Just wait for 1 minute. Just wait for 1 minute. I have changed the slide already. I think the question is visible now. Okay, thank you. Please try to answer. Okay, I think all of you have answered B, base excision repair. Let's see. So, uh, I think the question was quite clear for everyone. As which of the following mechanism will remove uracil? We are talking about a single nucleotide. So, the answer will be base excision repair. No question about it. So, how this base excision repair works? Let's look at it, the schematic diagram. Responsible for removing small non-helix distorting base lesions from genome. Okay. For example, uracil is deaminated and become guanine. Okay. So, what will happen then? This uracil DNA glycolase is another enzyme. This will move to the mutation site and it removes the unwanted base uracil. It will remove this unwanted, cause sorry. This deaminated C, because G should pair with C, right? Guanine should be paired with cytosine. If deaminated cytosine, that is uracil, is present here, so it is not forming any hydrogen bond. Okay, so that's why it is called an DNA lesion. Okay, one DNA lesion has been formed here, and so to repair this thing, uracil DNA glycolase will be present here, and it will remove the unwanted base uracil, because we need C here, not U, and it will produce one AP site. AP site means a purinic or a pyrimidine site. A purinic or a pyrimidine site means it will completely remove the nucleotide base and there will be one space here. As you can see, it will be creating one space here, and it is called AP site, a purinic or a pyrimidine site. That means there will be no purine or pyrimidine. So what will happen then? Epiendonuclease and phosphodiesterase will come here and they will cleave the epi site. Okay. So as you have already discussed in the previous class, this endonuclease, what they do, they just cleave the nucleotide base pairs at a particular site, at a particular restriction site. If we call about restriction endonucleases, here it is epiendonuclease. That means it will create or break the uh, bond, phosphodiester bond, and just remove this epi site from here, creating one gap. Okay, if a gap is there in the DNA, then DNA polymerase and ligase will get activated and they will add new DNA and seals the gap. Simple. DNA polymerase just add the new nucleotide required for here, that will be C at that point, and ligase will seal the gap, and that damage is repaired now. It's called base excision repair. It's a simple repair mechanism, because as you can see here, not much enzymes are required here, and it's just a mutation of single nucleotide base. Okay, this generally happens. That's why in the question also it is asked as uracil, because cytosine is deaminated and becomes uracil, and it is present at the place of cytosine, and it won't be creating any hydrogen bonds. That's why an lesion has been formed, and it leads to a purinic or a pyrimidine site, and that epiendonuclease or phosphodiesterase, AS means enzyme. Okay, it will just cut the phosphodiester bond and cleave the episite, and DNA polymerase and ligase will just add new DNA and seals the gap. So now the damage has been repaired, and it's simple base excision repair mechanism. I think everyone is clear with base excision repair now. Is it clear to everyone? Do you understand what I am saying? Thank you. Thank you for replying. Okay, okay. Let's move to our next question. Which of the following has the self-repairing mechanism? DNA and RNA, DNA, RNA and protein, only DNA, DNA and protein. Which one it will be? Please try to answer. Okay. So the answer is only DNA. Okay. Because there will be no question about it. Only DNA has a self-repairing mechanism because only certain DNA polymerases act in that way, and DNA has this proofreading mechanism for itself only, not any RNA or protein. They can't be repair itself. If a protein is damaged or RNA is damaged, it will be damaged. Only DNA has this DNA polymerases act in that way and proofreading mechanism for doing these things. Okay, let's move to our next question. The DNA polymerase involved in base excision repair each. Which DNA polymerase it is? Because in the previous slide we have discussed about how base excision repair mechanism happens, and there must be one DNA polymerase that adds the new DNA and ligase just seals the break. Okay, so here, as you know, there are set number of types of DNA polymerases are there. So which DNA polymerase is involved in base excision repair? Please try to answer. Oh, slides has not changed. Okay, let's see. So the answer is DNA polymerase beta B. It is involved in the base excision repair. As we have already discussed the base excision repair, I don't think we need to repeat it. Let's move to our next question. This is an alteration in a nucleotide sequence that changes a triplet coding for an amino acid into termination codon is nonsense mutation, mutagenesis, mutation, or mutagen. What it will be called? Okay, okay. Just for 1 minute. Just wait for 1 minute. I think the network is slow. Here it is. The question. Please try to answer. Okay, very good. I think everyone has answered. And answer is nonsense mutation. An alteration in a nucleotide sequence that changes a triplet coding for an amino acid into a termination codon. Okay, that will be nonsense mutation. Okay, what are the nonsense codons? Just look at this schematic representation. These point mutations can be like just for it will be C, DNA level, mRNA level, and protein level. Okay, if this TTC, it is the DNA sequence, it's transferred transcribed to AAG in case of mRNA level, and that leads to this amino acid lysine. Okay, in case of silent mutation, what happens? As I have already cleared about it, the third genetic code, if it changes, this T changes to A. So it won't affect for the whole triplet codon, and it will be the same amino acid, that is lysine. Okay. In case of nonsense codon, nonsense mutation, what is happening? If it is a codon like ATC and A is mutated to U, okay, A is mutated to U, then it will be called as UAG in the mRNA level. If it is UAG in the mRNA level, then it will be act as a stop codon for the translation process. Okay, so it won't be responsible for any amino acid, and the codon will be stopped there. So it might be happen like if the protein won't be formed, any complete protein will not be there, or any misfunctional protein might be there. Okay, this is known as nonsense mutation. In case of missense mutation, it can be of two types, like conservative and non-conservative. Okay, is my audio or am I audible? Okay, thank you. So this missense mutation can be conservative or non-conservative. So TCC is the sequence of DNA at the DNA level. If it replicate or transcribe to AGG, in case of mRNA level, it will produce arginine. Okay, arginine is a basic amino acid, base amino acid, basic. If this G is mutated to cytosine, guanine is mutated to cytosine, and it produces one amino acid called threonine, which is in polar amino acid. Uh, it is an polar amino acid. So it will be called as missense mutation. So basically, what is missense mutation? In this case, just a single nucleotide base changes, and that leads to change of the whole amino acid. Okay, so as you can see here, it might lead to different amino acid in case of TTC, but if it stands to C to G, then it leads to production of another amino acid or change of protein sequence. Why it is said as conservative and non-conservative? Because if there is one missense mutation which leads to, if it just for example, this TCC or AGG, it will code for one basic amino acid. It might change if after mutation also it codes for another basic amino acids. It means the basic basicity or acidity is not changing. That's why it's called conservative. The basicity or acidity is maintained. Okay, so and in case of non-conservative, if it changes, that like if because of mutation the amino acid change from polar to non-polar or non-polar to polar, or acidity or basicity of amino acid changes, that means it is non-conservative, it is not conserved, and it will lead to the protein it lead to changes in protein folding, protein function, and other cases also. Okay, because if because also you know that polar and non-polar amino acids act in a different way. It might be hydrophilic or hydrophobic, or it might changes the whole protein function also. Okay, understood? I think everyone is clear with silent mutation, nonsense mutation, and missense mutation. Specifically, we just get confused with this missense mutation. What is it? So it's nothing, just a single nucleotide change is happening in the triplet genetic code. It is called missense mutation, and that leads to another amino acid formation. Okay, I think everyone is clear. Let's move to our next question. Which of the following DNA repair mechanism is known as cut and patch mechanism? Options are photoreactivation, mismatch repair, nucleotide excision repair, base excision repair. Please try to answer. I think the screen is visible to all of you. Okay. Some of you have answered B, some of you have answered D or C. Okay, let's see. So the answer will be nucleotide excision repair. It will be C. How? Let's discuss. Generally, what happens in the nucleotide excision repair, there will be a cut and patch mechanism, like we have already discussed about this thing like pyrimidine dimer. When UV light is exposed to DNA, it generally forms a thymidine dimer. Okay, so what happens then? This UVRB and UVRC, they leads to with the help of one ATP molecule, they attaches the UVRBC complex to the damaged portion. UVRB and UVRC, they just bind at the damaged portion. After this, loading of UVRC to thymine dimer happens, and UVRD will cut the dimer. Okay, so there will be one cut. That means complete TT nucleotide has been removed. In the previous case, in the photoreactivation process, we have seen that this DNA photolyase, what it does, it just breaks this TT dimer. Okay, with the help of FADH, it just breaks this phosphodiester bond and produces monomeric bases. Understood? It produces monomeric bases in case of photoreactivation process. But in case of nucleotide excision repair, it completely removes the nucleotide by UVRD. And after that, DNA polymerase 1 fills the gap and DNA ligase seals the gap. Everyone is clear with it? I think it's very simple. Just pyrimidine dimer, they generally happens because of UV light damage to DNA. Okay, and after that UVRB, C and D, they are required for this nucleotide excision repair with the help of ATP molecule. This attachment of UVRB and C happens at this thymidine dimer. After that, UVRC or UVRD will cut the dimer. Completely it cuts and creates one gap. Okay, in the previous case, the TT bond has been broken in case of photoreactivation process. But here in this nucleotide excision repair, it has been removed completely, and after that it will be the gap has been filled by DNA polymerase 1 and DNA ligase seals the gap. Okay, everyone is clear with nucleotide excision repair? Let's move to our next question then. Why recombination repair system is called double strand break repair? This is the last question of our today's session. Please try to answer, then we will discuss. Try to answer this question. Okay, I got a diverse of answers like A, B, C, D. Great. Okay, so let's see why recombinational repair system is called double strand break repair system. So the answer is A, that is both strands are broken. both strands are broken. Okay, some of you have answered D, both strand act as template. So generally in the DNA replication process only, both strand act as template. One of will be the leading strand, another will be the lagging strand, no difference between them. Understood? So it's a general process of DNA replication where both strand act as template. In case of recombinational repair system, if we talk about, it is called double strand break repair because both strands are broken. Let's see how it works. So, if any exogenous factor or endogenous factors affect the DNA and double strand break happens here, it leads to production of single strand DNA tails, like here. Okay, single strand DNA tails has been formed, and break detected and 5 days and digested. This is just a breakage of DNA in the double strand helix. After that, this 5 strand edge has been digested for both the strands. Then what happens? Strand invasion. 3 days and invades homologous region of sister chromatid. So I think you can correlate with homologous DNA repair mechanism. Okay, homologous DNA repair mechanism. What happens? So this three days DNA strand will move with the homologous strand sister chromatid. Okay, it will move like this of homologous sister chromatid, and then D loop extension happens and fill in synthesis happens in the presence of DNA polymerase enzyme. After that, just simple ligation process happens and DNA breakage has been repaired. Okay. This recombinational repair system is used when both strands are damaged, such as when the DNA is broken. In such a situation, one strand can serve as a template for repair of the other. Okay. Some of you have answered D. So why it can't be D? In this situation, one strand can serve as a template for the repair of other, because both of the strands are already broken, so we can take one strand as a template to repair the other strand. That's why it's called recombinational repair. It is similar to the homologous repair system where both the strands depends upon their sister chromatids, homologous sister chromatids, and they follow the follow their sister chromatids for extension of the whole gap and to fill the gap, and at the end ligation happens in the presence of enzyme ligase and the breakage has been repaired. Okay, I think everyone is clear with recombinational repair system. Do you understand? Okay, thank you for replying. So, this is all for today. Thank you for attending the session. Uh, I think we have discussed about nucleotide excision repair, base excision repair, and certain damaging factors also, and how it is working, and how we can cope up with this repair mechanism. So this is the some sample questions I have collected from the site of NPTEL. Also, as you have already gone through this question, so you might be able to answer your assignment of week 2. I'll suggest you to read the lecture slides and attend the lectures properly so that you can get a clear understanding of breakage of DNA and repair system. Okay, you can ask any questions to me if you have any doubt. If you have any doubt, please ask me. Okay, this is all for today. Thank you.